Hello dear aspirants, welcome to GDC Class English and especially for the another video in Pharma Thunderstorm series, especially designed for all pharma competitive examinations. Yes, okay, I hope you subscribe this channel. If not, do it immediately because you're going to miss a lot of content which is coming in future itself. Okay, yes. So now in this video, I'm going to explain about plant hormones. What is that? Plant hormones. Before that, I just want to start with the basic, introduction of plant hormones. First of all, let me know what is hormone and why they're required. If you know this answer, you're able to understand what I'm going to explain in the upcoming slides. Okay, yes. So we know that for every biological function, for every part of our body, for every cellular process, we require a chemical messenger called hormones. What are that? We require a chemical messenger called hormones for every function in our body to carry out every process in our body, like every part of our body. Okay, there is an involvement of a chemical messengers called hormones. Without hormones, okay, let us consider growth hormone, okay, follicle stimulating hormone, right? And lactating hormone. There are so many hormones in our body. Each hormone have their own function, own cellular process, right? So which means you can see right over here, the plant, not only the plant hormones, when it comes to the hormones, okay, they are mainly meant for the coordination and balance between the organism life processes, whatever the process may be, to maintain the coordination and the balance between the life processes, right? Clear? So the function of hormone, it is a chemical messenger, but it is mainly for the coordination and the balance between all the process, all the biological process which are carrying out in the life cycle in the living organism, right? So the hormone, but where it comes to plant hormones, yes, plant is also a living organism, so it should also carry out so many processes such as a metabolism, right? Okay, metabolism, growth, reproduction, respiration. To carry out all this function, the plant also require hormones. That's the reason they call it as plant hormones, or they called as phytohormones. Plant hormones, or they called as phytohormones, even though this function is also same. Plant hormones, the phytohormones function is also same. Okay, yes, these also will coordinate the balance between all the biological process. They maintain, see, they coordinate the metabolism, they coordinate the respiration, they coordinate the growth. Clear? To carry out all this process, to coordinate all this process, the plant require, right? The plant require plant hormones are the phytohormones. Yes, they're known as, another word for this plant hormones known as plant growth regulators. Plant growth regulators. Yes, and coming to that, yes, ultimately the plants will grow. Simple terms, right? Simple terms. The plant require, plant need to be grow, and that's the reason. In order to regulate their growth, we require the plant growth regulators. That's the reason they are plant hormones, also known as PGR or plant growth regulators. Is it clear? Yes, simple word within a one single word. Plant hormones are nothing but the chemical messengers which are carried out all the cellular process, okay, all the biological process in the organism, okay, that may be metabolism, that may be respiration, that may be growth, or whatever it may be. Clear? Yes. Coming to that, let us see and let us start this plant hormones topic, by considering all the competitive examination, all pharma competitive examinations. Okay, yes, let us see. Yes, as I told you that plant hormones, also known as the phytohormones, that are chemicals that regulate the plant growth in generally. See, majorly in plant, what we need, plant should be grown enough to give the secondary metabolites, thereby fruits and thereby other essential things, right? So ultimately the plant hormones are mainly required for the and to regulate the growth. That's the reason they're known as phytohormones. Okay, phytohormones, and that are signal molecules produced within the plant and occur in the extremely low concentration. Do remember, the plant hormones, even though in our body, the human body also, we do synthesize our own hormones. Let us consider insulin, right? Insulin is a hormone which is, and then Langerhans islets is going to be synthesized, but ultimately it is going to be synthesized in our body when you're lacking, when your Langerhans islets, when your pancreas get damaged. That time only, at that instant only, we are going to get these insulin from the external source. But until that, Your body is synthesizing your own hormones, which means every living organism can synthesize their own hormones within their body, That's the reason you can see plant will synthesize its hormones within the plant itself, okay? And even though, but very low concentration, do remember they'll ask you, okay, very low concentration they can synthesize, very low concentration, okay? At very low concentration, they synthesize their hormones. But when it comes to the function, the hormones regulate the mainly cellular process, because a cell is a structural and functional element of our body. It can regulate all the functions. So that reason, this hormones will regulate the function. This hormone will regulate the cellular process. Thereby, if you regulate the cellular process, all the function, all the biological functions, right, will be regulated. That's the reason they are the chemical messengers. They give the chemical signals to the cells. Thereby, they will work according to the signals that they're obtaining from the hormones. Is it clear? Yes. And in the targeted cells locally, and when moved to other locations, yes, they will work on the targeted cells, as well as when they move to the other location also, they work as same as like that. Okay. And in other location of the plant, they affect the gene expression. So now coming to the what is the main possible, what are the most possible effects on the plant? Gene expression, transcription levels, and cellular division and growth. So majorly these plant growths or regulators or plant hormones, they affect the gene expression. Okay, gene expression and transcription and cellular transcription levels and cellular division and growth. So these are the majorly four mechanisms or four ways of affecting the plant life cycle by the plant hormones. When it comes to the classification, okay, how many are there? We do have only five plant hormones are there. Five plant hormones are there, in which again we have classified them into plant growth promoters or plant growth regulators or plant growth inhibitors. Okay, so based on the function we have classified them into two types. The first three, that is auxin, cytokinin, gibberellins, are plant growth regulators or promoters. They will promote the growth. Okay, we'll discuss. And plant growth inhibitors, the name itself will tell you inhibit the growth. They are inhibiting the growth of the plants. Okay, yes, they are nothing but abscisic acid and ethylene. And yes, from this, I'll again just we'll frame a very simple questions. Okay, yes, they'll ask you a very simple question we can expect from this classification is they'll give you four examples. In that, they'll ask you which one of the following is a plant growth regulator, or which one of the following is a plant growth inhibitor. Okay, so for that reason you should remember the classification as well. Okay, first three, ACG. Okay, ACG are the G means growth regulators. Okay, ACG, auxin, cytokinin, gibberellins are the plant growth promoters or regulators, except there's two. Okay, abscisic acid and ethylene, these two are the plant growth inhibitors. Okay, they'll ask you which one is an inhibitor and which one is a promoter. And among these five, only one is in a gaseous form. Only one is in gaseous form, that is ethylene. Okay, ethylene is a gaseous phytohormone which is synthesized within the plant. Clear? Yes. Do remember the classification. From the classification also we are going to get some important questions from your examination. Okay? Yes. So that is about the plant hormones introduction, which means plant hormones, they are also known as phytohormones or plant growth regulators. They are the chemical messengers. They are also known as chemical messengers because they regulate the functions of cells. They regulate the cellular process by sending the chemical signals. So they are known as chemical messengers. They are known as chemical messengers. Why they are known as chemical messengers? They send the signal molecules. They send the chemical signals to the cells. Thereby they regulate the process. That's the reason they're known as chemical messengers. They're known as chemical messengers. Okay. Yes, you know about they are available in a very low concentration. Okay. Yes. Now coming to that, at which stages? Okay. At which stage in generally we do have five types of plant hormones, but we should know at which stage of the plant life cycle they are going to involve or they are going to be synthesized. Okay, first one is gibberellin. Okay, gibberellin is synthesized or it is useful in the plant in the first and early stage, that is germination. Okay, it is in the germination stage. In the germination stage, the gibberellin is very important. G stands for germination. Okay, and whereas Auxin will involve in the initial growth of the plant. Initial growth. It is very, very important for the initial growth. Okay. Okay. Initial growth of the plant, and thereby cell development. For the cell development, cytokinin is important. Okay. Cytokinin is important. And but whereas abscisic acid will be useful for the dropping of the leaf and dropping and setting of the dropping. Okay, dropping and ripening of the leaves and the fruits. Okay, and lastly, ethylene is mainly for the ripening, mainly for the ripening. Okay, yes. So by this you should understand what is the role of the each plant hormone in each stage. So gibberellin is mainly important in the germination stage. It learned for the it is mainly important for the germination. It facilitates the germination. Okay, and where auxin which facilitates the initial growth of the plant, and whereas this cytokinin will helps in the cell development. Okay, and whereas the abscisic acid will mainly meant for the abscisic acid is mainly meant for the dropping. Okay, dropping of the fruits and leaves you can see here. Okay, and also for dormancy. Okay, and lastly, whereas ethylene, it is a last phytohormone synthesized from the plant mainly for the ripening of the fruits. Okay, so without ethylene no plant or no fruit is going to be ripened. Okay, if you consider bananas, right? If you consider the bananas in generally, okay, not only the bananas, what are the fruits? Okay, when we keep a raw or unripened fruits in one empty room, right? So when you close the door and when you come back after one day, when you open the door, you feel that gas, some special aroma you're going to experience that because of the ethylene is released from the plant. If it releases ethylene, that means they have ripened. Is it clear? So don't worry, we'll discuss each one in detail. Okay, clear. But before that, let us see the functions. Gibberellin mainly to stimulate the stimulates and breakdown of stored food to germination and sprouting of leaf. That's the reason it is involved in the germination. How it is involved? How it facilitates germination? It break down the stored food. Say in the seed, okay, in the seed we do have an embryo, right? Embryo, which means all the stored food, right? All the stored in the seed. If you consider it as a seed, it contains embryo, which is the stored food, right? Stored food for the seed for the future development. And this stored food, the stored food will be the stored food will be utilized for to develop a seedling through a hilum. See, in generally, if we consider this as a seed, right? In this, in generally, for every seed we do consider there is a point dot. Every seed contain a opening called hilum. Okay, and from this hilum only we are going to get the seedlings. Okay, so but here itself there is a part called embryo. In this, all the stored food which is mainly helpful, it is mainly useful for the seed development or seedling development for germination. Okay, yes. So, but what is the function gibberellin? This gibberellin will help to break down the stored food to germinate into a new seedling. A new seedling. There is a point, a minute opening is there in every seed that is called as hilum, which is mainly for the facilitation of water transport. Okay, water intake. So when the water intake is coming, when the water when the water intake has been taken towards this hilum, okay, it is enters into the stored food called embryo. Okay, so from this only we are going to get the entire food. Okay, entire seedling. Clear? Yes. So gibberellin will helps to break down the stored food and mainly stimulates the germination and sprouting. Okay, and whereas auxin, auxin Auxin will helps you in the cellular growth and cell elongation. Do remember, especially from this plant hormones, what type of questions we can expect? That is the main burning issue, right? Firstly, they'll ask a basic first question we can expect is which one is a plant inhibitor, plant growth inhibitor, and plant growth promoter. That's the first type of question we can expect. And second type of question, what's the major function of each plant hormone? So when it comes to gibberellin, what's the function? Where is maybe germination, okay, and cell elongation. When it comes to auxin, cell growth, cell elongation, okay, and when it comes to Cytokinin, the function is cell differentiation. Okay, the function is cell differentiation. Okay, and when it comes to abscisic acid, dormancy. Okay, dormancy is a function of abscisic acid, but whereas ethylene, the function is ripening. So you should remember the second type of question is, first type of question we can expect is what is what is the classification. Second type of question is we can expect what is the major function of each each plant hormone. And third type of question we can expect is plants to have a natural hormones and synthetic hormones. Plant to have a natural hormones and synthetic hormones. So, what is a synthetic hormone? What is the example for synthetic hormone? And what is the example for the natural hormone? So, these things also we should definitely remember, because based on that only we are getting a lot of questions in your competitive examination. Okay. Yes. So, cytokinin is mainly involves in the cell differentiation. Okay. Involves in the cell differentiation. And whereas abscisic acid will involve in the dormancy and dropping of ripened leaves. Okay, and ethylene is mainly for the ripening of leaves and fruits, as well as if it produces excessive of ethylene, it can also lead to the dropping of the leaves. Okay, so let us see each one in detail. Okay, let's discuss each one in detail. Come on. First one, auxins. Okay, auxins are the first plant hormones that are discovered. Very, very important. Okay, and also do not forget that. Okay, in this, at the end of this session, I'm going to give you 10 important questions which are already given in the previous paper. Okay, listen carefully, thereby you'll answer the questions. Okay, yes, auxins are the first hormones. Okay, what are the hormones that are firstly discovered? Auxins. Do remember, A is the first alphabet, so that A stands for first. First alphabet is A, and A is auxins are the first hormones that are discovered. Okay, and yes, I told you already, plants to contain natural auxins as well as naturally auxins and synthetic auxins. Do remember, natural auxins are nothing but IAA, okay, 4-chloroindole 3 acetic acid, okay, and IBA, indole 3 butyric acid. IAA, 4-chloroindole 3 acetic acid, okay, and indole 3 butyric acid are found to be the natural one. But whereas synthetic are, okay, NAA, naphthalene acetic acid, one naphthalene acetic acid, and 2,4-D, very, very important. This is a, this is synthetic and commercial auxin which is available in the market for the plant growth. Okay, 2,4-D, 2,4-D is a synthetic auxin available in the market. But when it comes to functions, we know that. Okay, see, first three are categorized into plant growth promoters. If you want to promote the growth, which is what you mean by growth, the plant should be grown, the size should be increased, which means there should be a two process. Ultimately, if it is a plant growth promoter, they should have only two functions. One is cell elongation and the one is cell differentiation. Okay, one is cell elongation and another one is cell differentiation. Yes, thereby only two cells are going to be formed now. So, cell elongation, cell differentiation. Thereby, from this again, this cell is elongated and this can be differentiated. Like how cells are elongated and differentiated to form a new tissue, right? Entire process going to be continued. Okay, and thereby a new tissue is formed and thereby growth is going to be achieved. Clear? Yes. Similar to cell elongation, which means the function of the auxin is cell elongation. Okay, and also the auxin supply from the apical bud suppress the lateral growth, of lateral buds. Okay, which means if yes, if the particular the oxygen is supplied, for the from the apical buds to to suppress the growth of lateral buds, clear. And yes, next, differentiation of vascular tissue. Yes, exactly. So how do you differentiate? In the plant trunk part, in the plant stem part, we are going to have a xylem and phloem. Okay, the xylem and phloem differentiation also can be done by this one, IAA. See, plant to have a two, in the plant stem part, there are a vascular tissue. In that, they have differentiated into two parts. One is xylem and phloem. How can you differentiate, and which one is responsible for differentiation from the xylem and phloem? Only one thing, IAA, that is auxin, is the only thing which can differentiate the xylem and phloem. Okay, xylem and phloem. Clear? And yes, that's one of the other function. And also they stimulate the root initiation on stem cuttings. That's the reason in the plant tissue culture, auxin is a very, very important hormone that is required, especially for the stem culture. Sorry, stem culture, okay, and root culture, stem culture and root culture as well. Because if you add this auxin to the any part of the plant in a suitable medium, it can initiate the roots. That's the reason root initiation is going to be done. Okay, and also auxin also various effects on leaf and fruit abscission and fruit setting and development as well. But main function is, do remember, cell elongation, and they suppress the growth of lateral buds. They suppress the growth of lateral buds, and also they differentiate the vascular tissue into xylem and phloem, and also they initiate the root. Okay, so that is about auxin. Natural are IAA, 4-chloroindole 3-ester acid, and phenyl acid, and IBA. Okay, and two important synthetic auxins are naphthalene acetic acid. Okay, one naphthalene acetic acid and 2,4-chlorophenoxy acetic acid. That means 2,4-D. Okay, clear. Very, very important. Do remember, these are the first auxins, first plant hormones that are discovered, and thereby natural, synthetic, and functions. That's all. Next, gibberellins. Very, very important. Okay, the name of this plant, the name of this particular uh, plant hormone has been written based on one, okay, skeleton gibbane. Okay, gibbane, because this particular gibberellins has been synthesized from the a disease called Gibberella fusicorae, and when we observe this Gibberella fusicorae, they contain, okay, gibberellic acid. They contain this, the gibberellins are produced from the Gibberella fusicorae. There is a fungus or a disease. Okay, it is obtained from the disease, but this Gibberella fusicorae is responsible for a gibberellic acid. In this gibberellic acid, we have found a skeleton called gibbane, and from that we have written this name as gibberellin. Gibberella fusicorae, gibberellic acid, gibbane skeleton, and yes, that's the reason they're known as gibberellins. Clear? Yes. And let us see. The group of plant growth regulators discovered by Japanese workers in the connection with the 𝙂𝙋𝘼𝙏 disease. Okay, actually, they have found this accidentally. They have found this gibberellins accidentally, which means in the 𝙂𝙋𝘼𝑎𝑛 foolish seedlings. Actually, especially when they have investigating the rice plant, okay, when they have investigating the rice plant, they have discovered one disease called 𝙂𝙂𝘏 disease. That is nothing but foolish seedlings. And while they were investigating, they got to know there is a one causative organism called Gibberella fusicorae. Okay, and when they are investigating on this Gibberella fusicorae, they got to know there is a one important and useful hormone called gibberellic acid, right? And yes, that contains gibberellin, and yes, from that only gibberellin has to be synthesized. Okay, and yes, in this particularly in this gibberellins, they have only 136 total 136 gibberellic acid are there, but only GA3 gibberellic acid 3 is the first gibberellin to be structurally characterized. Do remember, okay? GA3 is the first gibberellin to be structurally characterized. Okay, from the plants, fungi, and bacteria. From the plants, fungi, and bacteria. Clear? And yes, now it moving to the functions applications. Function applications. Okay, auxins, gibberellins, and cytokinins. These three are mainly promoters. Do remember, do not forget, do not confuse. These three are promoters. So ultimately, if they are promoting the growth, either they have only two functions: cell elongation, cell differentiation. That's it. Okay, clear? Yes. So increase the axis length of the plant, such as grape stalks. Okay, when it comes to applications. Okay, mainly for, if you see, up to the 19th century, the grape stalks, the grapes, the length, the particular grape setting is in a grapefruit, entire the grape stalk is there na, that stalk, okay, that grape stalk is there na, the entire fruits of grape stalk was very shorter. Okay, but when they have discovered this gibberellin, the grape stalk, now you can observe, around 100 centimeters. Okay, sorry, not 100 centimeters, around 10 to 15 centimeters length they can increase. Okay, clear? Mostly to increase the axis length of the grape stalks. Yes, they are useful, they are applicable, and also to delay the senescence and fruits, such as resulting in the market period is extended. Okay, mainly if you use a gibberellic acid, okay, the shelf life, or else we can increase the life. Okay, we can increase the life of the fruit, which means we can delay the senescence. Okay, we can delay the senescence, which means we can delay the aging in the fruits, thereby the fruit seems to be very fresh. Yeah, so mostly gibberellins are also used to delay the senescence. Very, very important. What are the points I've underlined? Those should remember. Okay, yes, very good. Okay, and also helps a fruit like apples to elongate and improve the shape. Yes, to be, to be in a good shape, because mostly gibberellins are the, they are mainly involved in the cell differentiation, right? Cell elongation and cell differentiation, mainly to differentiate. That's the reason, when the cells has been clearly differentiated into each and every corner of the particular fruit or stem, thereby they become a very good shape. Clear? To improve their shape, okay. And mainly gibberellins in the commercially in industry mostly, okay, they use this particular GA3 or gibberellins to speed up the malting process in the brewer industries. In the brewing, which means in the all the alcoholic industries, they use this gibberellins for the speed of the malting process, right? And also spraying the gibberellins increase the sugarcane yield, and also they can increase the length axis. They can increase the lengthening of the, okay, sugarcane. To increase the length of sugarcane also we require gibberellins, okay. And also they used to has in the maturity period in a young coniferous and promote the, yes, simple one point, promote the early seed production. They promote the early seed production. They promote the early seed production. And lastly, they promote the bolting. They promote the bolting, which means sudden growth of plant just before the flowering. Before the flowering, if you grow the plants, that will be a bolting. But this bolting process will be applicable in cabbage and beetroot. Cabbage and the beetroot. Clear? Yes, that's about gibberellins. In general, it is known as gibberellin has been, they have synthesized from a costum organism called Gibberella fusicoride, right? From that, we have synthesized a gibberellic acid structurally, right? Around 136 gibberellic acids are there, but only GA3 is the first gibberellin has to be synthesized, okay, has synthesized, okay. And yes, they have found this Gibberella fusicoride has been found in one disease called Bakane disease. Clear? It is discovered in the rice plant. Okay. Yes. You have seen these functions. Do remember, we are going to get the questions on the functions as well. Next is cytokinins. Cytokinins. You can see from the name itself, cyto means cell, kinins means differentiation. Cytokinin means cell differentiation. So that within the word we can say cytokinin means cell differentiation. Okay. And when it comes to the history or introduction, okay, Skoog and his co-workers observe a mass of cells called callus in tobacco plants. Okay, callus in the tobacco plants, and these cells proliferated when the nutrient medium contains auxins along with yeast extract or extract vascular tissue. Yes. Okay, first of all, when the Skoog and Miller, right? When the Skoog and Miller, when they have, when they are going to done a research, sorry, okay, when they are performing a research on a mass of cell called callus, okay, and they have observed, okay, they have interestingly they have observed this particular mass of cell was growing up, okay, in a nutrient media when it contained auxins along with yeast extract, but later they have identified an active substance responsible for the proliferation called as kinetin. Simple within one word. In generally, Skoog and his co-workers, they are performing an experiment on the plant tissues. Okay, so in that, a group of mass of cells called callus. Okay, this callus was growing enormously with the in the concentration, in the nutrient medium containing auxin as well as yeast. Okay, they thought that because of auxin and yeast, this part called callus was growing enormously. It was proliferating enormously. But later they have identified for the growth of this cytokinins. Okay, there is another one substance called, okay, kinetin is responsible. Okay, so this kinetin is known as a cytokinin. Okay, so the first discovered cytokinin is kinetin. The first cytokinin discovered is kinetin, and this kinetin does not occur naturally, but scientists later discovered several natural examples such as zeatin. Okay, see what happened here. In generally, when the Skoog and Miller, they have performing an experiment on the plant tissues. Okay, when they have discovered a callus. Okay, so in the nutrient media, they contain auxin as well as the yeast extract. Okay, the proliferation of a mass of cell is going to be done, but they thought that for the growth of this proliferation of the group of mass of cells called callus is done by auxins and yeast extract. But later, due to some biological reactions, okay, after some time, they have discovered a new compound called zeatin. Okay, so the zeatin belongs to the cytokinin. They have decided and they have concluded that this kinetin is a belongs to cytokinin, but it was not naturally available. Okay, due to the reactions between the oxygen and other yeast extract and the nutrient composition, it has been synthesized. But the naturally discovered or naturally present, okay, yes, first identified is kinetin, but naturally identified and naturally available cytokinin is zeatin. Okay, naturally identified or available cytokinin is zeatin. Do remember, okay, kinetin is the first cytokinin obtained, first cytokinin is discovered, but whereas zeatin is the first natural one. Clear? Yes, the natural cytokinin mostly available in the root apices. Okay, root apices and developing the shoot buds. Obviously, this function is cell differentiation. Okay, that's the reason they available in the root apices. Okay, in the root apices. Mostly in the root and shoot they are available. Mostly in the root and shoot they are available because they need to differentiate into different parts. Cytokinins are the things, cytokinins and the hormones, they'll decide whether it should be become a fruit, whether it should be become a leaf, whether it can be converted into a flower, whether it is become into flower bud or tendril. So this can be done by the cytokinin. Clear? That's the reason they present in the either shoot tip or root tip. Clear? Shoot tip and root tip. Because at this point only we are going to decide whether it become a root or whether it become a fruit or leaf. When it come, why they present in root and shoot? Okay, because in the shoot part only, we need to get the leaf, fruit, flower, right? Tendrils. Yes, so this can be divided by the cytokinin because it differentiate. Okay, cell differentiation is the main function, cytokinin. And in root also they are present. Why they present in the root? Okay, whether it's become a primary root, secondary root, tuber or rhizome, they can also divide it by the cytokinin. Clear? Yes. So let us see the functions. It is helps in the formation of new leaves exactly and chloroplast, and they promote the later shoot growth and advance shoot formation. Yes, that's the reason they present the shoot and root only. Okay, in the shoot, why they present the shoot? Because from the shoot only we are going to get the leaf, fruit, flower, right? Yeah. Yes. And when it and they also present in the root as well, because in the root only we are going to get the tubers, rhizomes, okay, everything. And also they help to overcome the apical dominance exactly, and also they promote the nutrient mobilization, which in turn helps to delay the leaf senescence. Okay, they promote the nutrient mobilization because cell differentiation has been done. That's the reason. Okay, they promote the nutrient mobilization from one part to another part. Clear? Yes. Let us let us summarize this one. Cytokinins, nothing but they are mainly involved in the cell differentiation. Okay, Skoog and Miller are the two persons who firstly identified this kinetin accidentally. Okay, accidentally when they are performing a callus. Okay, when they are performing the research on callus. Okay, clear. But the first identified is kinetin, but naturally identified is zeatin. Okay, zeamase. Okay, mostly present in the corn. Yeah. And yes, you know that the function is cell elongation. So for that reason, new leaves can be differentiated or formed by the help of cytokinin. Okay, chloroplast. Okay, and apical dominance can be done, and also lateral shoot growth and advanced shoot formations can also be done by cytokinin. Okay, so thereby we have completed plant growth promoters. Now we are moving to the plant growth inhibitor. Right, I told you already. Ethylene is the only hormone which is available in the gaseous form. Okay, which is available in the gaseous form. Okay, and yes, coming to that, what is the function mainly? See, the plant will synthesize. See, the ethylene. Okay, ethylene in generally released by the plants. Okay, if it releases the ethylene, that means the plants become ripened, the fruit become ripened, the part become ripened. Clear? Ultimately, it's one of the gases, only one. Okay, only one gaseous plant hormone present in the plants. Okay, yes, mainly for the ripening of fruits and tissues and undergoing the senescence produce the ethyl in large quantities. Okay, whatever the part, whatever the leaf or whatever fruit, if it is undergoes for the ripening or undergoes for senescence, that means they are releasing the ethylene. Clear? Yes. And coming to the functions, Coming to the functions, they affects the horizontal growth of seedlings and swelling of axis in dicot seedlings. Okay, mostly in dicot, no, axis of dicot seedlings. Okay, axis of dicot seedlings, and also, okay, and they affects the horizontal growth. Okay, and it promotes abscission and senescence, especially in the leaf and flowers. Okay, abscission and senescence. Okay, and also they enhance the respiration rate. Exactly. Okay, what do you mean by respiration rate? See, if this ethylene has been released from the plant part, okay, that means ripening. Okay, so ethylene should be expelled out from the tissues because the process called respiration, right? More the respiration, more the ethylene will goes out and more ripening going done. That's the reason you can see. Okay, okay, they enhance the respiration rate during the ripening of fruits because they enhance the ripening. In order to enhance the ripening, or in order to ripen the fruit or leaf, what they need to do, they expel more amount of ethylene from the fruit. Thereby they can easily ripen. Okay, and this phenomenon is known as respiratory climactic. And also they increase the root growth and root hair formation. They increase the root growth and root hair formation. Thereby they help the plants to increase their absorption surface area. Clear? And also, majorly, you should remember only one thing, that okay, the commercial the commercial form, or the commercial ethylene in the market was available as ethofone. Okay, ethylene regulates many physiological process, therefore widely used in agriculture. Okay, in agriculture, commercially they use ethofone as a ethylene. Okay, ethofone is a commercially synthesized or commercially available ethylene is ethofone. Do remember, we are going to get a question on this. Ethofone is ethylene plant growth inhibitor, and the plants can easily absorb and transport an aqueous solution of ethylene. Okay, so this particular thing, okay, which is available in the powder form, and generally, and they mix it up with the water and they spray, and thereby this aqueous solution, okay, the aqueous solution of ethofone and easily absorbed, okay, and release the ethylene slowly. Clear? That's it. That's it about ethylene. I hope you understand. And lastly, abscisic acid. Okay, abscisic acid. This function is mainly for a dropping. Okay, and dropping of leaves. Okay, dropping of leaves and fruits as well. Okay, it is also called as ABA. Abscisic acid was first discovered and researched under two different names: dormin and abscissin. Simple. Within the word itself, you can know the function. Dormin will cause a dormancy. Abscissin to cause abscission in the leaves, which means dormancy, abscission. Okay, thereby fruit leaving or fruit dropping, you can see. Therefore, the chemical property were fully known. Obviously, the chemical property fully known. In general, it acts as an inhibitory chemical compound that affects the bud growth, seed and bud dormancy, which means they inhibit the growth. They are inhibitors. Okay, so they inhibit the growth by dormancy. They create dormancy, thereby ultimately seed growth, bud growth, and bud dormancy can also be done. Yeah, and yes, it mediates the changes within the apical meristem. Okay, and causing the bud dormancy and the alteration of the last set of leaves to the product the bud covers. Exactly. And lastly, in the plants, yes, water stress. And do remember, it is a stress hormone. Do remember, abscisic acid is a stress hormone because it is mainly helpful for the plant in the stress condition. Mainly plant in the stress condition. In the plants under water stress, ABA plays a role in closing the stomata. Okay, simple. Okay, when the plant is under stress, okay, the plant may tend to lose the water from its stomata, thereby, okay, in that type of stress condition, which means most of the, when extreme temperature is there, right, immediately the plant will release a abscisic acid. At the time, this abscisic acid will close the stomata, thereby they will prevent the excessive loss of water in the stress condition. That's the reason known as stress hormone. Okay, stress hormone, and includes the seeds and to synthesize the storage the protein, and thereby they close the stomata. They provide, no, they prevent the loss of water as well as protein from the seeds as well. Clear? Yes. So these are the five important plant growth regulators or plant hormones or phytohormones. You know that in this water plant growth regulators and water plant growth promoters and water plant growth inhibitors. Okay. So by this image you can understand. Okay. Mostly, okay, for the Leaf maintenance phase, oxygen is observed. Oxygen is important because oxygen will promote the cell elongation. Okay. And whereas, okay, if the ethylene is released from the leaf, that means it is going to be ripened. Okay, it is going to be ripened. Okay, so ultimately, when they release the okay ethylene and abscisic acid, when they release the abscisic acid, ultimately they cause a fruit drop, leaf and fruit dropping. Clear? So I hope you understand clearly. If not, try to take a snap or take a screenshot from this particular table. It is clearly mentioned or clearly described. Okay, take a pause. Okay, and we have mentioned all the introduction, natural and synthetic and function, mostly on this only. Okay, mostly on this source, on this 4 points only we are going to get about the introduction, which means what the definition, and about which one is natural, which one is synthetic, and what is a function. Clear? So basing on this only we are going to get the questions. Okay, yes. Now first question of this series, or first question on this one. Okay, yes, plant growth hormone. I'm going to show you 10 important questions. Okay, I hope you're ready. I hope you understand. Okay, yes, do not forget to mention the right answer in the comment box. Okay, from the question 1 to the last question. First question: which was the first discovered plant hormone? I know you are going to answer the right one. You're going to pick the right one, but do not forget to mention in the comment box. Gibberellin, auxin, cytokinin, or abscisic acid? Which one? Undoubtedly, yes. You write auxin. A alphabet stand for first alphabet. First plant hormone is auxin. Yes. Take a big round of applause for yourself because you opted the right one. Okay. Second question. Indole 3 acetic acid is classified as either a synthetic auxin or natural auxin or gibberellin or cytokinin. You can eliminate two options because you are very smart students. We know that because okay, it is belong to the auxin. That's the reason we can eliminate synthetic gibberellin and cytokinin. But what is the right answer here? Undoubtedly, the right answer is a natural oxygen. Natural oxygen, right? Clear? NEA, Okay. Yes. Next. Next, Buccal disease of rice is caused due to excessive production of which one? Where you have seen this word of Buccal disease? Simple. If you cross-link the word, where you have seen the Buccal word, there will get the answer. Okay. I told you already. In the Buccal disease is caused by the Gibberella fujikuroi. From the Gibberella fujikuroi only we are going to get this gibberellins. We have synthetic gibberellins. That's the reason the right answer is gibberellins. Okay. Yes, very good. Good going. Okay, great going. Next. Which of the following is a synthetic oxygen? Which of the following is a synthetic oxygen? Okay, synthetic oxygen they were asking. Okay, synthetic oxygen. Okay, zeatin, ethofone, 2,4-D and ZA3. Okay, which one is a synthetic oxygen? Undoubtedly 2,4-D. 2,4-D. 2,4-D is a synthetic oxygen. Okay, synthetic oxygen. Clear? Very good. And next question. The phenomenon of apical dominance is mainly regulated by apical dominance. Apical dominance is mainly regulated by which one? Which one? Yes, undoubtedly the right answer is auxin. Apical dominance can be done by the auxins. Is it clear? Yes, very good. Next, ethylene is commonly used in agriculture for mainly agriculture. Okay, ethylene, which means ethofone. We already seen, right? Ethofone. Which one is right answer? Yes, for ripening of fruits. For ripening of fruits, we are using this ethylene. Okay, mainly for ripening. Okay, very good. Next question. Which hormone is known as a stress hormone in plants? I discussed just now before. Okay, which of the hormone is known as stress hormone? Which of the hormone is known as stress hormone? Okay, which one? Okay, undoubtedly the abscisic acid. Abscisic acid is known as stress hormone. Very good. And now the synthetic compound ethofone releases which gas inside the plants? You know already which gas they release? They release either oxygen, ethylene, carbon dioxide, or hydrogen. I know you're a smart people, you're going to answer the right one. Undoubtedly, ethylene. Ethylene is the right answer. Very good. Okay. Last but not least, the discovery of gibberellin is associated with which fungus? Either Aspergillus niger, Gibberella fujikuroi, Fusarium oxysporum, Penicillium chrysogenum. What is the right answer? Undoubtedly, You are going to answer the right one. 𝘂𝙧𝙎𝑃ℎ𝑒𝑐𝑜𝑟𝑖. Okay. And yes, last question of this Pharma Thunderstorm series. I know you are going to answer the right one. Gibberellins are widely used in the brewing industry because why? Gibberellins are used in the brewing industry because it it enhance the root initiation or speed up the malting process or delay the fruits ripening or promote the seed dormancy. What is the right answer? The right answer for this one is ultimately to speed up the malting process. Okay, yes, this is for today's Pharma Thunderstorm series. I hope you understood and hope you like this. Okay, and do not forget to mention how many right answers you have got from the 10 questions. Okay, and if you have any doubts or if you have any queries or anything to say, just mention or drop your message in the comment box and we'll go through all the comments. Okay. Yes, and also if you have any request from your side, if you want any topics to be there, if you want to mention, okay, if you want to mention any topic under this Pharma Thunderstorm series, do not forget to mention under the comment section, okay? And thank you. Have a great day. Jai Hind.